Here are the contents of my presentation. Let us first proceed to understanding what a cell cycle and a cell division is. A cell cycle is a set of events that occur in a cell as it develops and divides. A cell spends the majority of its time in what is known as interphase, during which time it develops, copies its chromosomes, and prepares for cell division. The cell then exceeds interphase, goes through mitosis, and divides fully. Cell cycle helps in renewing of damaged cells, the production of new cells from older ones, maintaining the total number of chromosomes, providing more cells for growth and development, repairing and controlling damages caused to the cells, and also helps in the survival of growth and living organisms. The interphase. The interphase period is the time between two cell divisions. The interphase period is the time between two cell divisions. During this stage, the cell does not divide. Instead, it absorbs nutrients and metabolizes them, grows and replicates its DNA in preparation for mitosis. There are three stages of interphase. The first one is the gap period or G1. It is where cell grows initially. Synthesis of protein and ribonucleic acid or RNA occurs, and then the mitochondria increases in number. The second one is the synthesis stage or S phase. It is where the DNA are synthesized, thus replicating the chromosomes in preparation for the next cell division. And lastly, the third one is called the second gap period or G2. It is where cell grows rapidly and the cell prepares for the actual cell division. Speaking of cell division, the interphase is followed by the cell division phase. These are found in the eukaryotic cells. There are two forms of cell division. It is the mitosis and meiosis. But in our presentation, we are going to shift our focus in the mitosis process. Mitosis is a biological process in which the original nucleus divides into two nuclei in two cells, each of which has the same chromosomes number as the parent cell. Prophase, metaphase, anaphase, and telophase are the four phases of mitosis. Let us tackle those four phases right now. The repeated coiling of chromosome occurs in the prophase. Those chromosomes are made up of two sister chromatids that are identical to each other because of the replication of DNA during the S phase, and those two chromatids are still attached at the centromere of the cell. The last occurrence is that the nuclear membrane breaks down in the prophase stage. The next stage is called the metaphase. It is where the chromosomes align at the equatorial plane, and then the spindle fibers from both centrosomes connect to each chromosome through its kinetochore. Metaphase is followed by the anaphase. In this stage, spindle fibers that are attached in the metaphase stage begins to contract and become shorter. The continued contraction causes the separation of the genetically identical sister chromatids. This causes the centrosomes to divide and produce two opposite poles with single chromatids. The last phase of the mitosis is the telophase. After creating separate poles, the chromosomes are now at the opposing poles of the spindle, and the microtubules disappear. Then, the two sets of chromosomes divides the nucleus into two, and begins to be surrounded by new nuclear membranes. This process is called the karyokinesis. After multiplying the nucleus and having new nuclear membranes, the cells are ready to be separated by the last process called cytokinesis. It is where the separation of the cytoplasm of those chromosomes occur. Now, we have a product of two identical daughter cells, each with a chromosome of 4 to 6, just like the parent cell. There ends our understanding in the process called mitosis.